Well, I'm not sure how long we have there, but thank you. I'll see what I can always pull from last year if that's a problem. Okay. So, neurotransmitters, let's talk about, well, let's start over here. What kinds of things could make an action potential go faster down the axon? Does anyone know? Yeah, myelin. You can increase your speed with myelin. And how else can you increase your speed? We're going to talk about the glial cells that are responsible for myelination at the end of the talk today. But does anybody else know how you can increase the speed of an action potential propagation down an axon? Pat, did you? I'm sorry? Enzyme? I can't. Enzyme. No, it's not an enzyme. Something to do with the size of the axon or the diameter of the axon. You think a bigger one or a smaller one would be better, make things faster? It's actually bigger. Okay, the larger diameter axons are going to have a faster propagation of the axon potential. And the way to think about that is that there's less resistance in the diameter of the axon. So if you had a really big garden hose, things don't go as fast, or things are able to go through faster. You're going to get more through without any resistance. Okay, so that's larger diameter axon. Okay, so increase the speed of the action potential, two different things. You can myelinate, or you can have a larger diameter axon. Okay, let's talk about neurotransmitters. I mentioned, does anybody know any neurotransmitters? No? Acetylcholine. All right, that's going to be a general neurotransmitter. So I'm going to categorize these. Let's talk about some general neurotransmitters that we're going to talk about during the course of the semester. In general, there's acetylcholine, A-C-E-T-Y-L, choline. Okay, and then another general transmitter that we're going to talk about during the course of the semester is going to be dopamine. Does anyone know what disease process dopamine is going to be important for? Somebody say Parkinson's? Yes, Parkinson's. We're going to talk more about dopamine. Dopamine is a general neurotransmitter. It can be either stimulatory or inhibitory, depending on the receptor that it binds to. Now, other kinds of neurotransmitters. There's going to be the major excitatory There's a major excitatory neurotransmitter in the CNS. Anybody know which that one is? That's going to be glutamate. So the major excitatory neurotransmitter is going to be glutamate. And then there's also a major inhibitory neurotransmitter in the CNS. Anybody know which one that would be? GABA, yeah. GABA is going to be your major inhibitory neurotransmitter. And as Dr. Boyle likes to say, GABA dabba do. Okay, now thinking about this, we're going to talk about it again when we get to Parkinson's disease, but increase in sodium causes depolarization to minus 50 millivolts. It hits the axon hillock. It generates the propagation of an action potential, which is the presynaptic terminal, causes the opening of calcium channels, calcium influx into the cell, neurotransmitters are released. Okay, neurotransmitters, let's say that a GABA was released here and bound to the cell surface receptor on the next neuron in sequence. What's going to happen inside this neuron? Are we going to depolarize or hyperpolarize? Any thoughts? Hyperpolarize, yeah. It's an inhibitory neurotransmitter, so that's going to stop the signal from going forward. Okay, but if this were glutamate bound, then you would cause depolarization and another action potential in the sequence. Does that make sense? Yes, no, sort of, maybe. Okay, and that's it in general. Thinking about synaptic communication between neurons in sequence. Moving on to neuron histology. Does anyone know what histology is? What's histology? Sorry? Tissue cells. Looking at it under a microscope, right? What's this thing actually look like? We're going to look at the basic neurons that we're going to talk about all semester, just different kinds. We're going to see that the function of the neuron has a specific structure that you should associate that function with. Okay, let's start with sensory. Okay, there's going to be a few things here, so got to kind of keep to the left-hand side of the drawing. And let's start with sensory neurons. Okay, your basic sensory neurons, so receiving information from the environment and bringing it into your central nervous system. Is that going to be an afferent or an efferent neuron? Right, afferent. Great. Taking something and bringing it in. That's an afferent. Okay, and when thinking about sensory neurons, there's going to be two basic things that we're going to think about. One's going to be somatosensory. We'll start with that. Okay, somatosensory. What does that word mean? Does anyone know? Feeling? Okay, feeling. Anything else? What is that German word? What? Somato. What's soma? Body. Yeah. So any kind of sensory information that's coming in from your body. Okay, and that's going to be a little bit different than other areas. We can also talk about, so somatosensation is what I'm feeling with my body, and then we have a couple of special senses that we can consider too, and that's going to be vision and olfaction. Okay, the kind of neuron that's responsible for conveying somatosensory information to your central nervous system looks different than the kind of neuron that relays visual and olfactory information to your central nervous system. Okay. And like I said, structure is going to follow function. So if we consider the somatosensory neuron first, the structure has a name. It's called a pseudo, P-S-E-U-D-O, unipolar, P-O-L-A-R. It's a pseudo-unipolar neuron. Okay, your pseudo-unipolar neurons are responsible for bringing in somatosensory information to your central nervous system. Okay, your pseudo-unipolar neuron, for the most part, when we talk about the spinal cord, you'll see resides in should have got some water. It resides in the dorsal root ganglia. We'll talk about more on that. So when we talk about the dorsal root ganglia, first day of class, does anybody remember if that's gray matter or white matter in the PNS? Gray, yeah, that's going to be your gray matter. So your pseudo-unipolar neuron has a specific structure, and I'm going to try and draw it down here. So we're going to have like the dendrites, kind of a little fuzzy little top there, and then coming off the dendrites, have an appendage, and then a little stalk, and coming down again. Okay. Oh, and then this is going to be, this is your axon terminal. So here's your dendrites. Let's label that. So these are your dendrites. Okay, here's your cell body or your soma. And down here you have your axon terminal. Okay, pseudounipolar neurons, the neuronal cell body, specifically the soma, as we're going to see, resides in the dorsal root ganglia. The dendrites go out toward the periphery, and then the axon terminal is going to be coming in to the next neuron in sequence, depending on the pathway. Okay, the next, so somatosensory neuron uses the somatosensory information coming in uses a pseudounipolar neuron. Vision and olfactory information coming into the CNS uses a bipolar neuron. Okay, your bipolar neuron. And vision and olfaction are just special senses that we'll talk about later in the semester. And your bipolar neuron looks a little bit different than your pseudounipolar. You still have your dendrites here up top. Okay, and then you're going to have appendage. You're going to have your cell body. So there's no stalk coming off. You're just going to have your cell body right in the middle. And then coming down, you're going to have your axon terminal. This is your bipolar neuron. So bipolar neurons are used to receive visual and olfactory information coming into the CNS. As I said before, these are your dendrites here. This is your soma. And then here's your axon terminal. Okay. So even though both of these kinds of neurons receive sensory information, they look different depending on what information they're receiving. Special senses use your bipolar neurons, and somatosensory information comes in through your pseudounipolar neuron. Questions? Okay. So that's your sensory afferent information. Let's look at the next one, and just to divide these, I'm going to go ahead and put some dots to make the next section over. We talked about sensory information being afferent, so motor information, what's that called? That's considered efferent? Afferent? Efferent. Motor information coming into the system or going out of the system is efferent. Okay, so these would be the kind of neurons. So the neuronal cell body is going to reside in the motor cortex and then travel down, and you're going to find out, to the corticospinal tract to hit the next neuron in sequence. Okay, these are your basic kind of neuron is a multipolar neuron. They're your basic multipolar neurons. Okay. Well, these basic multipolar neurons look like my stuffy friend. Okay, the standard, the axons, the dendrites, axons, axon terminal. Okay, so to draw that, I'm just going to try and do what we saw before, with the axons coming here off the top. The dendrites coming off the top. There's your soma with your nucleus. And the motor multipolar neurons have a really, really long axon, and then the axon terminal. So this is going to be your dendrites are here. You're going to have a really long axon. And then axon terminal. Okay, the multipolar neurons that are part of the motor system have characteristically long axons. Okay, and those are your basic motor efferent neurons, what they look like, multipolar, the long axon, and then moving over, if you want to make one more section, squeeze one more category in here as far as your basic neuron is concerned. The last category we're going to talk about are called interneurons. I-N-T-E-R neurons. Okay, and interneurons are, think of them as connector neurons. They're connectors and they're only in the CNS. Okay, so you have your basic sensory neurons, pseudopolar, bipolar, motor neurons, multipolar, and then your last are your interneurons. And there's two different kinds of interneurons. First is your A-N axonic. A-N-A-X-O-N-I-C neuron. And as the name suggests, these are just neurons. They look like your multipolar neuron without an axon. Okay, so to draw that, I'm going to try and make it look like my multipolar with the dendrites in the cell body. As it turns out, one of these dendrite-looking things actually functions as an axon. You just can't tell it's an axon by looking at it. Okay. So that's your anaxonic neuron. And it's nothing more than a connector neuron. We're going to talk about these anaxonic or connector interneurons when we get to the reflexes and the spinal cord portion of the course. The other kind of neuron is another multipolar neuron that acts as a connector. And the multipolar neuron looks pretty close to the multipolar neuron used here in the motor system. The only difference is it's going to have a very short axon. So it has a characteristically short axon compared to the motor multipolar. So if I were to draw that here, I have my dendrites coming out of the neuron. Okay, and there's my cell body. Okay, I'm going to have an axon, and then the axon terminal. But this axon is going to be a short axon. And that those multipolar neurons with the short axons just function as connector neurons in between other neurons. And they're not necessarily part of the motor or the sensory system. They're their own brand of connector. Questions about that? These are just what these things look like. Okay, we're going to be talking about all of them this semester. Okay, great. Let's go ahead and take a 10-minute break.